All right, good morning, everybody, and welcome to the Friday Informatics Institute Power Talk series. I'm very happy to introduce today's speaker, Dr. Chindo Hicks, who is Professor of Genetics and Director of the Bioinformatics and Genetics Big Program at Louisiana State University Health Sciences Center. And he's well known to us, been very much involved with our bioinformatics data science activities, including being a mentor and committee member for our hackathons. And I'm very happy to introduce his talk, Harnessing Bulk and Single Cell Omics Data for the Discovery of Biomarkers in Lung Cancer.
Okay, so thank you very much, Amy, for that introduction and for giving me the opportunity to share some of the work that we are doing together. So today I'll talk about harnessing bulk RNA-Seq data and single cell RNA-Seq data for the discovery of biomarkers in lung cancer. The title in the announcement was just cancers, but I'm going to mostly focus on the lung cancer and then a little bit with COVID. But the approaches that I'm going to be discussing or describing here are basically universally applicable to other types of cancer or even to other types of diseases where you have similar data. Originally, we have been working a lot in breast cancer and prostate cancer, but recently we started working on the lung cancer and then also working a little bit on the COVID-19. The work I'm going to describe today is mostly preliminary, and part of this work actually was also motivated by the discussion I had with Jake in trying to establish a collaboration between the Informatics Institute and our group here. So a lot of the work, the information I'll present, some of it is preliminary, but some of the work has progressed. The hope is that this information, when shared, could probably lead to some interest in collaboration between the Informatics Institute and our group. And see. What's this one? Okay. Seems like the slides are not advancing somehow. Okay, so basically the work I'm going to present also is not an individual work. It's basically done by our group here. So these are the members of the bioinformatics program here. We are fairly a small group, but we have grown. At least we are now 3 full-time people and 1 postdoc student, and then we have some master students who will be joining the program. So we have Dr. Chindo here, who is working on the bioinformatics and computer algorithms, and then we have Aditi, who is interested in who's a bioinformatics analyst, and David, who's a Ph.D. student. So in terms of physical location, we are located here at the LSU Medical Center in the Clinical Sciences building. And here at the bottom here are the different aspects of the data that we work on. We work on a lot of microarray data analysis, RNA sequencing, exome sequencing, whole genomes, and single cell sequencing. And also we have done quite a lot of work in methylation. And then we do the other bioinformatics stuff like data management, as well as the functional analysis. So I'm showing this information, as I mentioned before. We are part of the CCTS, which means we are a family, we are a member of the UAB family. So we do actually support also bioinformaticians or individual basic researchers in the partner networks. So for example, we have recently supported the people at Auburn University to get funding from the American Cancer Society and also the people at Tuskegee. So if you are a basic researcher and the investigators or the bioinformaticians at UAB are swamped, we definitely will step in and help out with those data analysis. So today I would like to talk about basically I'm going to give a little bit of background on our research domains. And then I'm going to talk briefly about integrating genomics data to infer the association between lung cancer and COVID-19. And then I'll also give a pilot study on integrating genomics data to infer the association between COVID-19 and lung cancer in smokers and non-smokers. This is basically to set the stage to when we are going to go to single cell integration of single cell data with the bulk RNA sequence data for biomarker discovery in cancer. And then I'll close with the future research directions and obviously some questions and some suggestions. And then some opportunities that are available here at UAB. At not UAB, but at the LSU, but also at UAB. So in terms of the research domains, basically, we are not restricted to a particular disease. We do bioinformatics and computational genomics, essentially meaning that we are open to collaborating with everybody who has bioinformatics needs, or someone who is interested in methodology development and other things like that. So we work a lot in try to understand the structures of the genomes. So this is mostly the work of genome mapping and things like that for genome analysis, understanding the biology of the genomes. In this case, basically, we are talking about the cancer genomes and other common human diseases. And then understand the biology of the disease and advancing the science of medicine. We do have interest in the working in the area of drug discovery, but we are a small group, so we can only do so much. But definitely we are open to work with the others who are working in that sphere. So we have some capabilities. And then like everybody else, we are very much interested in the, building predictive models, especially polygenic risk scores. We are interested in that. That is integrating different types of data from these domains, but also using some of that data and integrating it with genotype data to kind of refine the prediction models for polygenic risk scores. So for the people who are interested in like basic researchers, again, I would like to emphasize that we function as a research program and an education program, but we also provide the services. And these are the pipelines, basically meaning that these are areas where we have the technology and the capabilities to assist in terms of data analysis for the investigators in the partner network. And some of the things that I'll be talking about today are going to involve some of these analysis. We analyze individual data sets, but then also we integrate different types of data. For example, we have been integrating genomics data with the epigenomics data for quite some time. And now we have decided to move into integration of genomics data from bulk tissues with the single cell data, and eventually maybe integration with the single cell with DNA methylation data, but that will be a subject of discussion at another time. So why are we looking at what's the need for genomic data integration here? So this is basically, this is not generated by me. This is actually generated by the Broad Institute, but it simply highlights the complexity of the human system that we try to investigate. There's the issue of discovering the genes, but then there's also the issue of which cells actually are driving the disease. For example, cancer by definition simply means that it's uncontrollable growth of cells. So which genes are actually perturbing those cells, that becomes an issue. So that means for today, I'll talk about, I'm going to focus on the line cancer here. As I said, we have also worked on breast cancer, but for the sake of time, I'll focus on line cancer here. So line cancer is one of the leading causes of disease death worldwide. Probably the leading cause worldwide in terms of the death rates and everything. Even in the United States, and here in Louisiana ranks among the highest states in the US with the instances of lung cancer. But lung cancer also, those who have been working on the COVID-19, like maybe Jake, maybe Dr. Smino, who have worked on the COVID, there is also evidence that individuals who have lung cancer actually do have very poor outcomes when they are impacted by COVID. So that's actually what motivated our research to start looking at the lung cancer and the COVID-19. So, and you can see right here at the bottom here, there are many different types of cell types that are involved in lung cancer, but are also shared with the COVID-19. So what I'd like to focus on today is look at this. So this is where the integration. So this is just the basic design of the experiment. So if you look at on the right side here, this cartoon would be the US population. And then we have, out of the US population, we have people with COVID-19. And we have people who are normal meaning that they are not affected by the disease. And also here, then we have people who have lung cancer. Unfortunately, now we do not have a data set that has people who have both COVID and lung cancer in the same individual. So we don't have that data. So the data we will look at here will be more like a correlative studies where we are simply establishing the hypothesis or by association. So that's basically the idea here. So a little background about this. So this is a cartoon showing, for example, if we were just looking at lung cancer, where we have sequenced the tumors, the normal tumors, and then of course the normal tissue and the lung tumors. I'm not going to go into all the aspects of data analysis. It's pretty straightforward for RNA-Seq data analysis. But the principle is that we have two subpopulations. We have people who are not affected, and we have people who are affected. And we do the sequencing, we do the alignment, and basically we perform, we do all the mapping to the transcriptome, and we do the quantification of the sequence reads. We do data cleaning, and we go for downstream data analysis. So for example, here we would be looking at, we are looking at just data quality control between the two groups, the cases and the normal, and basically we remove the data that is noisy, and we have a clean data set, and we compare the two groups, and we find which genes distinguish those groups. And then we can go on to making some predictions, for example, looking at the survival analysis and things like that. So that is the simplified version of RNA-Seq data analysis in a simpler version. But also, there are cases where now we have a situation whereby instead of looking at the bulk tissue, that's the tumor tissues, we are actually looking at the single cells. So again, the process here would be, in this case, the cells themselves will be treated as the subpopulations. So we have a population of cells, but within that population, there are subpopulations of cells that we want to find. And we can also do the control. So the experiment will look simply like this, where you have sample of single cells. You do all the wet lab experiments. You get to here as a bioinformatician here. The first thing, of course, is to classify these cells, annotate the cells, and then perform the data analysis. Data analysis pretty much is the proceeding like in this cartoon here. So this is the process of data analysis. You get data from the sequencing platform. You have the genes and you have the cell types because you have already annotated the cells. So you can just compare between the cells and the controls, and between the normals and the controls, which is essentially what is happening here. Or alternatively, you can just do data reduction by looking at using a principal component analysis and look at the different principal components to see which cells are driving that. After that, you have annotated, you have clustered the cells, that's the annotation. You do differential expression, you find the cells, and basically you organize this, the cell types, and then basically you organize them into cell clusters. So there you have an independent data set analyzed based on single cell. You can always go further than that, do cell-cell interactions, and do the pathway analysis. So to find out which pathways are driving those cells, or which pathways are picked up by those cells. So that's basically if you are just working on single cell versus single cell by itself and the bulk RNA-Seq by itself. But our interest is to try to find out, okay, what is the relationship between the single cells? So we say cancer is uncontrollable growth of cells. But what is the relationship between those cells and the tumor itself? Because the bulk RNA sequencing is coming from tumors. In other words, if you can figure out that, then you probably can be able to start designing models for predicting which cells are the drivers of the disease. Or alternatively, if you can find out which cells are the bad cells, then you can decide to say you're going to use single cell sequence data to use for, say, drug screening or finding the targets. For example, if we knew which cells were there and we could design drugs that can target those specific cells, so probably there may be less use of chemotherapy, which then involves killing all the cells. So that's basically the principle there. So this is just a cartoon showing that the principal components, once you have analyzed the single cell data, you organize it, you do the annotation, and then you can have that information. So that's the background between those and these two. So I'll get back to now looking at the integrating single cell. Actually, I'm first going to look at the integrating bulk RNA-Seq from lung cancer and the bulk RNA-Seq from COVID patients. So again, we have the population like this. So we have COVID people and we have the normal and this. So we have done analysis based on this design where basically we take the lung cancer people, we compare them to the normals. And we take COVID, we compare them to the normals. The sources of data for this lung cancer is all lung adenocarcinomas from the TCGA. And the COVID-19 is mostly from GEO. So here we are basically looking at which genes actually can be can give us the information or provide insights about the association between lung cancer and COVID-19. We are not stating that COVID-19 causes lung cancer. We're simply saying that people who have lung cancer perform poorly clinically when they are impacted by COVID-19. So we are saying that what does lung cancer and COVID-19, what do they have in common? So that's the idea here. So we did data analysis basically here. The goal was to characterize the landscape molecular crosstalk perturbation between COVID-19 and lung cancer, and to identify the molecular networks and signal pathways associating the two diseases. So this is just by association, not one is causing the other or anything like that, right? So the testable hypothesis here was that the genomic alterations in the transcriptome in COVID-19 and the cancer-affected lungs could identify a signature of functional-related genes associating COVID-19 with lung cancer. So the first criteria here, like in most of other diseases, because the gene expression can be disease-specific, so the first criteria is to make sure that the bulk RNA-Seq we are using from COVID is from the lungs, and also the same applies to the RNA-Seq data we are using from for lung cancer, as well as the controls. So the second hypothesis was that that COVID-19 and lung cancer have shared gene regulatory networks and signaling pathways. So that's the first criteria. So the foundation of that basically is the analysis type. So we have 52 COVID samples and 52 controls, and we have 594 samples from TCGA on the lung adenocarcinomas and the COVID. And so we analyze the two data sets separately. We integrate them to find the genes that are shared by the two groups. So this is basically, this paper was recently published actually. So we find that the genes here in the intersection are the genes that are actually associating the two diseases. The genes outside the intersections are the genes that are unique to each one of them. Then after that, we simply take the most significant genes from this group here in the intersection to see how they behave. So here we're just using unsupervised analysis, the hierarchical clustering, to find out which genes, how do they behave in COVID and how they behave in lung cancer. So we find that, and then we basically just run a gene regulatory network here, where we simply find which genes are upregulated and which genes are downregulated. In this case, red upregulated and blue downregulated. So these are only the genes that are associated with the two diseases. So that's so that was the idea to kind of establish the hypothesis that there's association between COVID-19 and lung cancer. Then we basically extended the same problem because now we have some hypothesis that is working. So we extended the same problem to say, okay, now we would like to see, what about if we look at COVID-19 and lung cancer smokers and non-smokers? The idea then being that smoking is one of the most well-supported risk factor for lung cancer. And therefore, we wanted to look at the biological mechanisms driving these associations in these two subpopulations. Are they the same or are they different? In other words, the association between COVID-19 in lung cancer smokers is it the same as the association between COVID and lung cancer in non-smokers? So we recently finished that analysis, and we found that again, we find that no, there are actually some genes that are just unique to smokers and the non-smokers. And then we also see that there are some genes that are common to both smokers and the non-smokers. But we are interested in genes that are unique to each one of the lung cancer patient groups. Then we take these genes here, we match them with the COVID-19, and we find that there are some genes, specifically these genes here and these genes here, the genes we are interested in, genes which associate lung cancer in non-smokers with COVID-19, but they are not in smokers, and genes which associate smokers with COVID, but they are not in non-smokers. So that was the, so that's something that we have done, and we have finished that. We have also done the pathway analysis, and the manuscript is in preparation for submission for that. So now we establish the foundation. So we say, okay, that the problem becomes much more complicated. So now we go to the cells, bulk RNA-Seq versus the single cell sequencing. Here again, the idea is that even though cancer initially we just use the bulk RNA-Seq, which is essentially meaning that we are sequencing the tumor genome, but there are many different types that drive the disease. Some of them may not be driving the disease, but some of them are actually driving the disease. So that's the idea here. So the idea is that we can see that, okay, we can analyze the data and find which genes are associated with lung cancer, but then which cell types do they actually pick up in lung cancer? So that's the first part here. So here we first start with only lung cancer without COVID. I'll come to COVID a little bit later, but we start here. So we say we have bulk RNA-Seq from lung cancer, but we also have single cell data from lung cancer. Now we are going to try to correlate the two and find out those genes which actually drive lung cancer or associated with lung cancer in bulk RNA, how do they behave in the single cells? So that's the idea. So we take the two data sets. So the first step again is to do this analysis here, analyze the single cell. So we have bulk RNA-Seq data here, which we already analyzed. We are also going to do validation because this is just bioinformatics using public available data. So we have to do validation. So we have 1,067 genes that are associated with the, so there is a typo here. It's supposed to be 1,067 genes, 667 genes that are associated with the, that have been confirmed as the immunomodulatory genes. And those genes are also involved in cancer. So that's the first step. So we have this data analyzed and we have this data here for validation. But then we go to look at single cell data. So the first order of the day again is to actually annotate the cells from RNA-Seq data and characterize those cells. Once we have those cells, then we can do we can compare their expression values between them, but also to the controls. So we find those genes and we organize those genes. Once we organize those cells, we can then do this. So for example, here we have the genes and we have the cells. So all these are the cells that are involved in lung cancer. So if we wanted to do clustering of all the cells using the cells in the columns as the population, so we can organize the data like that, or we can just do individual cell types and create heat maps for individual cell types. That is also fine. And then we correlate that data with the data from bulk RNA-Seq data. What I mean by correlating simply means that we have gene expression for those genes specifically in bulk RNA-Seq data. We also have the expression values in single cell. So we can generate the behavior of those genes. After that, we can do two things. One is once we have organized the cells like this, and we organize them based on the heat maps, we can also calculate a risk score across the cells. Essentially means aggregating the scores across the different cells to find out which cells pose a higher risk, and which cells pose a lower risk. So basically, to do that, basically we have to organize the data like this. In other words, you can see the stem plots here are simply showing that the mean values, the mean expression values, and here at the x-axis, you can see different cell types. And then here we have the scores that are computed from the gene expression data. Now, these genes here, and these cell types here, are only based on the genes that we have identified, as those genes are also found in the bulk RNA-Seq data. So that's basically how this heat map comes about. So once we have done that, we can see, okay, we can calculate that index and say lower score, a high score. This is basically very similar to, for example, if you are doing a prognostic prediction where you say which ones have the probability of dying, or which cells are, for example, in this case, if you wanted to find out which cells are driving the disease or are driving the clinical outcomes. So that information from clinical outcomes, again, we can get it from bulk RNA-Seq data. So that's the, so once we have done this, the next part then is validation. So we find which genes and their mean expression values in individual cell types. So the next thing is to say, are these genes actually involved in immunology of cancer, or immunomodulated genes in cancer? So then basically we take the genes from here, and we basically use a Circos plot, and we plot these genes here. So we can see that this is just a Circos plot plotted against their expression values here in the middle here. And then this is just annotation, the color code showing annotation of what those cells are involved in and what these genes are involved in, and also which pathways they may be involved in. So this you can do it at the individual cell level, or you can do it by aggregating the cell to find genes that are basically controlling the events in all the cell types. Because the TCGA data gives us the opportunity to use the also somatic mutation data, we can now go back and say, okay, we found these genes here. What is their status in terms of somatic mutations? Because we already have information on these genes. So from here, we can find out which genes are somatically mutated. And if they are somatically mutated, what is this frequency of their somatic mutations, and what is the nature of their somatic mutations? So then all you have to do is pretty much make an Excel spreadsheet, go back to the file with mutations, re-record those genes, do the quantifications by selecting the genes that have somatic mutations, and then you quantify the somatic mutations per person. We do not have information on somatic mutations in terms of single cell data. We only have somatic mutations from bulk RNA-Seq. But the idea here is to try to find out these genes that are involved in which are perturbing the single cells. Are they actually driver genes? So driver being defined that because the cancer development and progression is driven by somatic driver mutations. So at that point, you take those genes and you basically create a cartoon like this. You find the genes that are somatically mutated, and of course you basically find out their color codes. Basically, what is the distribution of those somatic mutations? For example, are they SNP variants, are they indels, insets, deletions, frame shift, and all of those things. And you basically characterize them based on this. If you have germline mutations also in here for these genes, which you can easily get from the GWAS catalog. So you can also incorporate that information here, because all you have to do is this gene, say for example, P53 has all these mutations, but also you can add information on germline mutations. Now, why is that important? That is important, for example, if you wanted to do to incorporate like mutation information and gene expression data in terms of doing polygenic risk scores, so that you can refine the risk scores in terms of the risk scores are not only predicting risk, but they are also linking them to the disease itself by incorporating mutation information. So that's the so that's how we are integrating the two data types, the bulk RNA-Seq versus the single cell sequencing. So what about if you come now to COVID, right? So as I mentioned, and as the others they have shown in the literature, that the reason why people who have lung cancer are performing badly for COVID-19 is because they have shared the mechanisms. So lung cancer here, if we see COVID is attacking the lung cancer is also attacking the lung. So for us, because we are not a wet lab, we are basic researchers, the first criteria we have to do to generate the hypothesis is to ask the question based on the genomics data from single cell sequence analysis and the RNA-Seq from single cell sequence analysis in both groups, is to find out the, because we've already shown that there's an association between the two diseases from bulk RNA. So now when we go to single cell, we ask the same question, which is, are there any cells that are involved in lung cancer based on single cell data that are also involved in the COVID-19, also based on single cell data? So the first thing is to look at a single cell data and look at finding the cells based on annotation. Are there any cells that are here but are also in COVID-19, or vice versa? You can start with COVID and also look at the lung cancer. So once we have found out that, the idea there is because things start getting complicated. So this is pretty much more like a data reduction approach, right? So that means then we can focus the analysis on the cell types that are common to the two diseases. So in this case, for example, we can see that the macrophages are here in the lung cancer, but they are also in COVID-19. The T cells are in COVID-19, they are also here. So once we establish that, we can analyze the single cell data. From this side here, from what I just talked about, we already know that we can integrate lung cancer with the single cell. Bulk RNA-Seq with the single cell RNA-Seq. Similarly, we do the same here. We can integrate the RNA-Seq with the single cell. So if we zoom in on the cells that are common to both diseases, essentially the cells that we hypothesize that these may be the ones driving the events, so the association between the two. So if we analyze those data sets, just looking at those subtypes, then we end up with a heat map that will look like this. First, of course, we are comparing the differences between the COVID-19 and the controls. Same thing we have done with the lung cancer with the controls. But in this case, we are looking at which of the genes that are involved in lung cancer are also found in COVID-19. So in this cartoon, for example, here, we can see that if you look at COVID-19 cases and controls, so the color codes here showing which ones are the cases, which ones are the controls, and then also the cell types. As I mentioned, we are looking at, for example, the alveoli, macrophages, because they are in both the diseases. And then we look at their expression levels, and we also look at their annotation status. And we can look at they are behaving separately. This is a simplified version of the cartoon. Obviously, the best would be to have another cartoon side by side, which is also showing lung cancer, or just everything combined with the lung cancer. But obviously that poses the challenge of presentation. So that's the so that's basically how we are doing that. We can then go further and look at what are the cell types, and again look at which genes are involved in here, and which genes, what is the cell status between those genes. Then after that, pretty much, so that's basically what we are doing in terms of doing that. So as I said again, this is something that we have been working on recently, and mostly after my meeting with Jake when he was visiting New Orleans, I think about 2 months ago now, that we are trying to generate a hypothesis that can we actually look at cell-cell communication. But before we can go to cell-cell communication, we have to see how do these cells actually relate to the phenotype of that. So in this case, it was much easier to first work with lung cancer and COVID to see what is the interaction. And part of it is this is not only lung cancer and COVID, but many of you guys have also heard that people who have pre-existing conditions kind of perform badly clinically when they are impacted by COVID. So essentially what I'm saying that this approach or this model does not apply only to lung cancer and COVID-19. It would be applied to obesity. It would be applied to anything that you are interested in. So going forward, basically, our interest, particularly the collaboration with the Informatics Institute, is to try to work in the business of maybe, so in this case, we see that integration of single cell and bulk RNA-Seq is a fairly powerful approach for discovering the biomarkers that are driving the events in a particular disease, or when we are dealing with comorbidities, like for example, someone has a chronic disease like lung cancer, and then they are impacted by another disease like COVID-19. So, so essentially, going forward is how to develop not only traditional bioinformatics approaches like simple differential expression analysis, but I think we are moving in the era of computational systems biology, where we basically have to model the systems. So with that, basically the future in terms of collaboration with the Informatics Institute, what we are trying to explore is to look at can we actually extend this kind of approaches to modeling cell-cell communications. So that's the basically that's how the future looks like. Whether we'll be able to do that, depending on how we garner the pilot projects that can support that and support those kind of research endeavors. So with that, so that was my closing, but I also wanted to point out at the end of this presentation that we do have some opportunities here at LSU. We have a fairly good master's program in bioinformatics. It's called the MS Biomedical Sciences Bioinformatics track. So we are currently actually enrolling into these programs. And so far we have a couple of students who have expressed interest in joining this program. So yeah, so I think I'll stop there and get some input or suggestions or comments, anything.
Well, thank you, Dr. Hicks, for the great presentation. And we do have some time for questions. If anybody has any questions or comments, feel free to put them in the chat, or you can also unmute yourself. I have a question. You're you had mentioned that that you were talking about different that you had talked about cancers in general, and that you were focusing more on the lung cancer. What are some other conditions that in which this model might apply, or that you've looked at already?
Actually, the model can also apply to, there are actually quite a few studies, actually, there are studies on, say, for example, cardiovascular disease. There are people doing that. And also obesity. There are also people doing that, looking at different cell types that drive things like obesity and things like, not colon cancer, but obesity and other metabolic diseases. So, for example, for those who are interested in other areas that are not cancer, you can actually use the TalkMed, which also has genomics data, which is basically under the National Heart and Lung Blood Institute. Is an equivalent database to the TCGA, but it's just based on the cardiovascular diseases. So you can actually replicate these same approaches to that. Or if you want, you can also replicate the same approach using brain tumors. There's the Human Brain Atlas, which also has the genomics data, as well as the single cell data. And if you wanted to do how different cell types of the brain, say, eventually how they communicate. For example, if you are studying Alzheimer's disease, that is also feasible to do that, and those data sets are also available in the NIH.
Other questions or comments?
If there are no questions, I have a question too. So, so I would like to ask Dr. Smino there, but he's muted.
No, I'm not.
Okay. All right. So one of the challenges that we have is, because I know that you have some work going on in the image network and things like that, or some kind of clinical phenotyping. So one of the things going forward that we would like to do this is to test some of these models in clinical applications, but I was wondering, we do not have that capability here, but I was wondering whether going forward, whether this is something that we can tap into the UBright. I don't know if UBright has genomics data as well.
So UBright has whatever data that you bring to it. And it also has access to I2B2, which has genomic data. But otherwise, I don't know what data sets. People bring their own data sets, and I suppose they maybe make them shareable and discoverable. But it doesn't have any, any large data sets that I know of. If Jake's on, maybe he can comment on that. I don't see him listed. But yeah, I mean, it's a platform for sharing data, and we bring I2B2 with its genomic, the genomic data it has, which I guess is really from the Alabama Genome Health Initiative, HGHI. But you said you were talking about clinical applications. So yes. What do you mean by clinical applications?
So what I mean basically is to correlate some of this data with clinical parameters, or validate them that if they are the predictions we are seeing from genomics data from this kind of bioinformatics analysis, do they correlate with the clinical parameters?
Okay.
Yeah.
All right. So that's not a clinical application, but yeah. So you can certainly do that. But we only have genomic data on a subset of patients. That's the AGHI. Not all of those people are actually patients at UAB. So we may have their genomic data and not have any other data.
I see.
All right. Well, if there are no other questions, I want to thank you, Chindo, for this presentation and contributing.
If I may.
Yeah,
Sorry.
sure. Go ahead.
I tried to submit a chat, but I didn't know how to actually post it. So I wanted to ask about what are considered the control tissues for lung. What is considered normal under the situation that there are many other conditions that can infect the lung, aging, death by pneumonia or asthma, and other injuries in life experiences. So how do you select what's considered normal?
So in this case, what is considered normal will be either it's adjacent to tissues from the tumor, which is essentially the definition that the TCGA has used, because those are paired samples. Or in some cases, we have, like, for example, in COVID, normal basically meaning that these will be disease-free people, basically meaning that they didn't have COVID, but those lung tissues were also extracted from people who died from other causes, but not from COVID.
Okay, thank you.
Any final thoughts, comments, questions? Well, if not, thank you very much for the excellent presentation. I hope you can join us for the next seminar. Everybody have a good weekend.
Yeah, sure. Thank you very much.